focusing on this because what's happening at the venial level, right? Why is venous hypertension causing this problem? What's really quite interesting is the capillary has a very simple endothelial lining, but the post-capillary venial, where the capillary turns into a venial, where all the trouble is. Trouble is not in the artery, it's not in the capillary, where oxygen is exchanged and sugars exchanged and amino acids. It's in the post-capillary venules. That's where the problem is. That's where the lesions form around. It has a different structure. It has another space around the endothelial wall called the perivascular space. This is what actually happens. White cells don't just go chomping on the blood-brain barrier. They are invited in, right? So it's a very elaborate process. First called tethering, then rolling. So this is the white cell that kind of gets picked up by the edges of the endothelium, and then they start rolling along the endothelium, then they actually attach to it, and then they go through. They're invited through the tight junction. They're not just blasted through, they're invited through, to get into the parenchyma. Here's a simpler drawing of the same thing, but it's a different author. So the rolling, bringing in the blood vessel, coming in through the tight junction. This is the perivascular cough-moving area. What's interesting about this, so here is the blood vessel. We have all of these leukocytes in the blood vessel, while the tissue outside the blood vessel is entirely normal. Right? So these guys aren't just warring through the blood-brain barrier. They're sitting here basically very much like in the chronic venous disease in the periphery that Dr. Bergen was describing in his paper. Another picture of the same thing. You can look at the blood-brain barrier in terms of gene expression. So there are certain genes that are expressed by the blood-brain barrier, so these are the left side columns are normal, and the right side are in MS. So in general in MS, all of these factors that control endothelial permeability are abnormal in MS. This is a lousy slide, but this marker, this is normal, and this is MS. This marker is also significantly elevated compared to MS. Zonulin is the molecule that controls endothelial permeability. The main marker of opening endothelial tight junctions is this molecule called zonulin. Well, zonulin is quite interesting. First of all, it's abnormal in MS, so you can measure zonulin in concentration of blood, normal, SPSS, RMS, in remission, so significantly elevated in zonulin, in MS. So zonulin is a pretty interesting molecule. It's very late development. We don't have it in other primates. Where am I going with this? Zonulin is also the controller of permeability in the gut. And what activates zonulin in the gut? Gluten.
Say it again. Say it out loud.
Come on, Mom.
So, gliadin is the actual molecule. Gliadin plus glutenin is what we call glutenin. The primary cause of abnormal intestinal permeability is excessive zonulin production, which is stimulated by gluten. This is biomarkers of gluten intolerance, MS versus controls. So they are significantly elevated in MS patients. So this is a case for being on a gluten-free diet. While I'm talking about diet, which MD should never do, the data on vitamin D actually makes it into neurology journals. Shocking, right? So you all ought to be on 5,000, 10,000 units of vitamin D. Okay, so now I want to put it all together. So so what's killing the oligodendrocytes? Well, so it's decreased perfusion, decreased diffusion. But what's going on? So I want to throw out to you a way that together. If you have in the venule reduced flow and or obstruction of flow, such as the leukocytes, perhaps what's happening is causing a shunting of blood around the area of the abnormal venule. So now, because the blood shunting around it, the oligodendrocyte is not getting its normal amount of perfusion and diffusion. It's essentially in a stagnated state, and that's why you get the lesions around the venule, and why you can put the whole picture together by saying venular disease is causing poor perfusion, stagnation in the brain. That's what's killing the oligodendrocytes and causing the debris that is then brought in to clean up the myelin debris after the fact. Thank you.
What was that keyword? Gluten? So any questions before we move to our panel discussion? Yes?
No, Mark, sit down.
Well, I have a question about this gut business. It's kind of interesting because iron is the uptake and regulation of iron is also controlled in the gut. And the problem is that there's almost no way to get iron out of the body. And the only way really is blood. And of course, women have the ability to get rid of iron a little bit better than men from that perspective. But there are
Delicately put.
there are some people who say that, having blood money or basically go and donate blood once a month or something like this, that has been demonstrated in people who have leukochromatoses to actually be very useful. It is a way, it's probably the only simple way other than using desperoxamine as a means to try to reduce iron in the brain. And that's not even being proven to work effectively in animals, let alone humans. So the key issue here is what's happened to iron regulation in the gut. And I'm not sure that Zamboni continue to follow up. What happens in terms of measuring the amount of abnormal iron in the blood in the form of transferrin, in the form of hemosiderin? There's a number of forms of iron that can be measured for like $10 chemical studies in the blood. What's happening to this group of people that Adam says 30% of these MS people have high iron content? They have that breakdown. Whether or not it's caused by shunting of the blood around it, I think it's just lack of blood flow around it, actually, based on Euland Good's data. What role does this gut abnormality play in controlling iron in these people? It would be very nice to do some of these simple blood studies to correlate in those individuals the iron content as seen in the blood with this gluten intolerance. This might be a very interesting area to look at. What role does the gut play?
Thank you for your comment, Dr. Yankee. Do we have any other questions before we? Yes.
Yeah, thank you. That was a very elegant presentation, well described. I wanted to ask what the likelihood of recovery is of either oligodendrocytes or axons if they're only partially, they're not completely apoptotic. No.
Yeah, that's known. So there are oligodendrocyte precursors that live in the brain, and it's known that once the oligodendrocyte dies, these progenitor oligodendrocyte cells come in and they do start remaking myelin. So a lot of the pharmaceutical industry is focused on how to stimulate remyelinization. So far, it hasn't worked, not because the oligodendrocyte precursors are not turning into oligodendrocytes and starting to make the myelin, but because the underlying problem is still there. So in this case, I would say the answer is the new ones that are trying to be made, they're going to be dying too if you have poor perfusion. And if you have a stagnant brain, they're going to die too. But there's a lot of effort on trying to get remyelination, and there is remyelination from these progenitors.